Hello, dear student. Today our topic is the scientific method or biological method. As shown on the screen, think like a scientist, record like a scientist, reflect like a scientist. It means that in case of biology and our daily life, we think about scientists, so solve the problem. So the scientific method or biological method. Science derived from Latin to know, way of asking and answering question about natural phenomena. Pseudoscience, false science. Science does not investigate the supernatural or study morality, religion. The very first step in biological method is observation and research. Observe the world using your sense, sight, sound, taste, touch, smell, and research your problem using internal or books or journal or any evidence from the environment. As you know that observation may be qualitative and quantitative. Qualitative is observed but not measured, while quantitative observation can be observed and can be measured, like length, weight, etc. So, the next step in biological method is a hypothesis. Hypothesis is the prediction about the problem that may be true or false. A scientist put forward a suggestion about the biological problem. As we give example about malaria. Malaria caused by plasmodium, or plasmodium is the cause of malaria. That is the example of hypothesis. The next step in biological method which is attached with the hypothesis is deduction. And we use if and then. And for example, if sunlight has an effect on plant growth, then plant will grow larger in the sun because it is needed for photosynthesis. Another example, if plasmodium is the cause of malaria, then all malarial patients must have plasmodium in their blood stream, in their blood content. So we know, if we compare hypothesis and deduction, a hypothesis is a suggestion about the problem, while deduction is the put forward a condition, and we use if and then. The next step in biological method is experiment, which is too much important. Experiment is performed on the basis of deduction. As we know that if plasmodium is the cause of malaria, then all malarial patients must have plasmodium in their blood sample. Test the hypothesis with a step-by-step procedure. It is clear and detailed, so another scientist can replicate your experiment. List all material. In experiment, we put forward two groups, experimental group and a healthy group. In experimental group, all the disease organism or animal are present, while in a healthy group, which act as a standard, which are normal, that will be compared with the experimental group. As example, measure the plant growth, amount of light is changed for each plant. The next step in biological method as experiment is further explain control group, healthy group or control group. As discussed earlier, the group sample that is used as a standard for comparison. Okay. Next, we collecting a data, qualitative or quantitative data can be put forward. Information observation collect during the experiment. Analysis. The last step is analysis. Okay. Thank you very much.